Hello guys, welcome back to my channel. Today we are going to solve GATE 2022 Biotechnology NAT and MCQ type of questions. So let's proceed. So first question is that circular plasmid has three different but unique restriction sites for enzymes A, B and C. When enzymes A and B are used together, two fragments of equal size are generated, and when enzyme C creates fragment of equal size only from one of the fragments generated by those cleaved by A and B, the plasmid is treated with a mixture of A, B and C and analyzed by agarose gel electrophoresis. The number of bands observed in the gel is. For say this is a circular plasmid. So now this question states that enzymes A and B are used together, two fragments of equal size are generated. So if we treat this particular plasmid with A and B together, then two bands or two fragments are generated. And these two fragments are of equal size. So if we consider that 6 kilo base pair is the length of this particular plasmid, so after treating with A and B, or after treating with this particular plasmid with A and B, the fragments will be of 3 kilo base pair each. So for consider A cut here and B cuts here this particular plasmid, and this side will be 3 kilo base pair and this side will be 3 kilo base pair. Next statement is enzyme C creates fragments of equal size only from one of the fragments generated by those cleaved by A and B. For say this is one 3 kilo base pair band and or fragment, this is again one 3 kilo base pair band. So now between these two only one fragment will be cut by C enzyme equally. So this 3 kilo base pair after being fragmented by C, it will be in 1.5 kilo base pair in size, and this again will be 1.5 kilo base pair in size. So if we treat this particular plasmid with A, B and C, what will be the fragments are? That is, one is about 3 kilo base pair and another one is of 1.5 kilo base pair, and again another one will be 1.5 kilo base pair. So in this case the number of the fragments will be 3, right? Now the question is that the number of the bands observed in gel. That means these 3 fragments are analyzed by agarose gel electrophoresis, and the number of the bands observed in the gel will be. So for say this is the agarose gel electrophoresis chamber or gel electric chamber, and here the larger fragments will be on this side and the smaller fragment will be this side. And for say this is 5 kilo base pair, after that 4, then 3, then 2, then 1, then 0. These are all in kb, and here are the wells where the DNA samples will be loaded. So the 3 kilo base pair band will be observed here. 1.5 kilo base pair will be observed in between 2 and 1 kilo base pair region. As the number of 1.5 kilo base pair bands are 2, but these bands will be showing as one band, but the band size will be thick. But in this case the 3 kilo base pair band will be in 1, so the band will be thin in appearance. So in this case the number of the bands observed in the gel will be 2, this one and this one. Next question is MCQ type of question, and it states that the overall stoichiometry for an aerobic cell growth is 3C6H12O6 plus 2.5 NH3 plus O2 is equal to 1.5 CaHP OCND plus 3 CO2 plus 5 H2O. So how we will do this? Let's see. We all know that in mathematical formula, that is LHS is equal to RHS. This side is LHS and this side is RHS. In LHS, the number of carbon is 3 into 6, that is 18, plus here no any carbons are available. Next is hydrogen. Hydrogen is 3 into 12, that is 36, plus 2.5 into 3, that is 7.5, so it will be 43.5. Next is for oxygen. Oxygen is 3 into 6, that is 18, plus 2 is equal to 20, and last one remaining is nitrogen, 2.5 into N, so N will be 2.5. Now in RHS, the number of carbon will be 1.5 into A. So 1.5 into A plus 3C, 3. So in LHS, the number of carbon was 18 is equal to 1.5A plus 3. So A will be 18 minus 3 divided by 1.5, which will be 10. So this was about the carbon, that is A value. Next is B. B is for hydrogen. So hydrogen is equal to 1.5 into B, 1.5 into B plus 5 into 2, that is 10. So in LHS, the value of hydrogen was 43.5 is equal to 1.5 B plus 10. So B will be 43.5 minus 10 divided by 1.5, which is 33.5 divided by 1.5, which is almost 22.333. Next is for oxygen, that is 1.5 into C, that is 1.5 into C plus 3 into 2, that is 6 plus 5 into 1. So 5. In LHS, the value of the oxygen was 20. So it will be 20 plus 1.5 C plus 11. So C value will be 20 minus 11 by 1.5, that is 9 by 1.5, which will be 6. And the last one is for nitrogen. Nitrogen was denoted as, nitrogen is denoted here as D, and nitrogen value is 1.5 D because 1.5 into D. And in LHS, the nitrogen value was 2.5 is equal to 1.5 D. So D value will be 2.5 divided by 1.5, which is equal to 1.6666. So now we have to see with this particular A, B, C, D values. So first option is saying that C9 and C is for A. So C is here 10. So this will be not right and this will be again not right. So C10 and C10, here these two options are with C10. Next is B is for hydrogen and B value is 22.333. So H22.33. O is for 6. So O will be 6, O6, and N is with 1.667. So the option D will be right answer for this question. So these are for the today's lecture. Hope this video will be helpful for you. Keep watching, keep supporting. Thank you.